Hello, my name is Majd Aris and I'm a senior scientist at Cell Signaling Technology. In this short video, I would like to introduce a new technology developed here at CST called IntraSeq, which will enable you to quantify proteins in your single-cell RNA sequencing assays. And towards the end of this talk, I will show you how you can characterize the underlying molecular mechanism of your favorite phenotype all in one experiment. If you'd like to download a copy of this poster, look for the links in the description below the video. While single-cell RNA sequencing is a powerful technique for studying the heterogeneity of cells, it does not provide any information about protein expression level, which is essential for understanding the functional state of cells. As a solution, we introduce IntraSeq, short for intracellular protein and transcriptomic sequencing, which is a novel technology developed by CST designed to revolutionize single-cell biology with an efficient and simple experimental workflow. The IntraSeq method enables the concurrent measurement of RNA, surface markers, cytoplasmic proteins, nuclear proteins within individual cells. And by leveraging CST's renowned antibodies, this method also enables the study of signaling pathways at the single-cell resolution. This technology was validated by CST in the 10x Genomics single-cell kits. The IntraSeq protocol is a 4-step straightforward immunostaining protocol. The first step requires fixing your cells with methanol overnight. At this point, you can store your cells in the freezer for a week without impacting the RNA in your cells. The second step involves incubating the cells with the SC block, which is a buffer mix that was optimized tremendously at CST to guarantee a robust readout for both RNA and protein in your single-cell dataset. At this point, the IntraSeq antibody conjugates are added to the cells for an overnight incubation. Finally, the cells are washed 3 times before being loaded into a 10x Chromium platform and processed with the 3' single-cell kits. This protocol requires only 1 hour of bench work spread out over 3 days. In this figure, we show how IntraSeq does not impact the heterogeneity of your sample. Here we took live peripheral blood mononuclear cells and processed half of them using the IntraSeq protocol. As you can see, the UMAP on the left looks exactly like the UMAP on the right, which presents the IntraSeq processed cells and live control sample respectively. The percent cell distribution was also very similar in both conditions, showing that the IntraSeq technology preserves the heterogeneity in PBMC. Here we show the feature plots displaying RNA expression of a specific target in the left panel and the protein expression of that same target in the right panel to assess the quality of the IntraSeq technology. For example, EBA RNA is expressed in a cluster of cells on the left of the UMAP, and you can observe that the protein expression of EBA is also detected in the same cluster as well. This cluster is also highly expressing S100A9 as well as CD68, both at the protein and RNA level. In most cases, you can observe that the protein expression is more uniform than the RNA, which displays one of the strengths of the IntraSeq technology by offering a more accurate representation of the target expression at the protein level. We also have many additional feature plot figures for other targets such as TCF1, NF-AT, FOXO, NCAM, ILOS, to cite a few. This shows that IntraSeq can display a very robust RNA and protein signature, especially when they are tightly correlated in single-cell datasets. However, we also find that RNA does not always correlate with protein levels. This graph displays the Pearson correlation coefficient between RNA and proteins for many targets. In certain cases like CD8, NCAM, and AIF1, the correlation is high and close to 1. When it comes to measuring post-translational modifications, this correlation between RNA and PTM is close to 0, showing a lack of correlation. This is somewhat expected since these modifications happen at the protein level and cannot be inferred from RNA expression alone. This is also a prime example of how IntraSeq can be used to uncover missing information in single-cell RNA-seq experiments and offer new biological insight at the post-translational modification level. In this example, we show how IntraSeq can facilitate the identification of cell states that would otherwise be difficult by analyzing RNA alone. After selecting CD8 positive T cells from PBMCs and reanalyzing the data, we found three main clusters that we labeled as naive CD8 cells, memory, and effector CD8 cells. Using the intraseq antibodies to measure protein expression, we found TCF7 to be highly expressed in the naive state of T cells, which corroborates with previously published papers. On the other hand, TBX21 and phosphocreb-serine 133, they were upregulated in the effector differentiated state. This is a great example where analyzing RNA level for these targets does not show a clear picture of the different cellular states, showing that the intracellular protein and PTM readout from an intraseq experiment offers a new perspective when identifying cellular states. To test whether we can accurately measure post-translational modifications using the famous CST signaling pathway antibodies, we inhibited Jurkat cells with wortmannin and LY294002. These drugs are highly selective inhibitors that target phosphatidylinositol 3 kinase or PI3 kinase. The red cluster on the right of the UMAP represents the drug-inhibited cells, whereas the blue cluster on the left is the controlled Jurkat cells. We decided to measure AKT phosphorylation level since AKT is downstream of PI3 kinase and impacted by these drugs. We found that total AKT remains the same in both control and drug-inhibited cells. However, phospho-AKT serine 473 levels dropped drastically only in the drug-inhibited Jurkats, showing that the inhibition was successful. The same pattern is seen with phospho-S6, which is downstream of AKT. This data offers a proof of concept showing that intraseq can measure PTMs in a single-cell assay. Similarly to the Jurkat experiment, we stimulated primary T cells with PMA and ionomycin to promote phosphorylation levels since they are generally low in primary cells. Once stimulated, the cells were split in half, where one of the half was treated with phosphatases to dephosphorylate residues on proteins. This data shows feature plots of phosphostat1 serine 727 and total STAT1. And as expected, phosphostat1 levels were high in the stimulated primary T cells, whereas phosphatase-treated cells had very low levels of phosphostat1. We can also observe a similar pattern with phosphocreb-serine 133. This data also shows that PTMs can be quantified in primary cells in addition to cell lines. In this last figure, we cultured PBMCs and stimulated half the sample with lipopolysaccharide for 3 consecutive days. This is an example of how you can use intraseq when you have two conditions in a heterogeneous and complex sample. And one way to analyze this data is by generating heat maps to show how phosphorylation levels of STAT3 serine 727 are impacted before and after lipopolysaccharide treatment. This is an example where you can determine which RNA or protein targets are correlating with these changes in phosphostat3 in each cell type. Since STAT3 translocates to the nucleus following phosphorylation and acts as a transcription factor, one could determine which of these targets are directly impacted by phosphostat3 by analyzing ChIP-seq datasets, for instance. Once you determine the promoter regions bound by STAT3, you can deconvolute the molecular mechanism in each cell type for your stimulation, inhibition, knockout or knockdown, or whatever conditions you may have. This process can start by measuring surface, intracellular, nuclear protein, as well as post-translational modification, all of this while quantifying RNA in single cells. Intraseq can also be a great screening tool to understand which signaling pathway and RNA targets are impacted in different cell types and help guide your research as to where to look. This technology can be employed as a great hypothesis generation tool when trying to understand the molecular mechanism in different biological conditions. This concludes our poster. If you are interested in implementing Intraseq in your lab, you can get in touch with your local sales representative or download a copy of this poster using the links below this video.